Hey everybody, welcome to a journal club. We haven't had one in a little while. We had some snafus in the earlier one, so my apologies for that. But we are back online. So this is Oliver Medvedik from Life Extension Advocacy Foundation and the rest of the crew. And we are going to be presenting not from Cooper Union, which is under a pandemic lockdown, as everything is still kind of quarantined in New York City, just to give you a little bit of a situation report from where I'm from. So here in New York City, things are loosening up a bit. The world is, obviously everybody knows the situation. I work at Cooper Union, which is a college, and there's a research lab there. Normally I'm at the Canbar facility, but we're trying to put in protocols in place to get people safely back in. So in the meantime, everything is virtual. So right now I am in my bedroom / library, which is overloaded with many books. This is where we're going to be doing journal clubs for a little while. So before I jump into the journal club, I want to basically give a bit of a shout out to Victor here, who is raising funds for a worthy project in collaboration with Longevity. So he's got is it a Kickstarter? Is it GoFundMe? What is it, Victor? I want to promote this because you guys really need the funds. Victor, can you unmute yourself?
Hold on, technical problem here. Let me try and unmute him.
Well, while Victor's unmuting himself, basically, we're going to be posting a link. He cannot unmute, but I'm going to be posting it on my Facebook page, and I think we're going to post it somewhere.
Yeah, he's unmuted now.
On a Leaf website. And this is to help Victor with his research projects he's right now on the West Coast. I don't think he can leave. But he's going to be working on a project there. So I wanted him to kind of mention the details because I don't have that page in front of me, but we are having some slight technical issues. Victor has been electronically gagged. Oh, he's
hello.
There we go.
Can you hear me?
Yeah, perfect.
Yeah, good. I am fundraising in order to work on a longevity-based project studying biomarkers for many people that can measure aging. So for the next week and until the beginning of June, I will be having this fundraiser to achieve that goal. So I hope everybody can help out in this situation.
And the work is going to be taking place where?
Here in Bay Area in Novato.
Oh, Bay Area. So Bay Area being in San Francisco, right?
Yeah.
All right. Very cool. So San Francisco is not having an earthquake right now, correct?
Fortunately not.
Okay, good. All right, excellent. So we're gonna have that link posted. So I'm gonna just head off to the paper right now. Everybody else who's listening in, please do donate to Victor's campaign, as I will be chipping in some funds for a very worthwhile cause. Okay, so on to the paper. What are we going to be discussing here? Well, Victor's working on biomarkers. This paper's on biomarkers, so how apropos, right? It's like we almost planned this. So the paper that just came out, it's I think it's still going through bioRxiv preprint. It's still undergoing, I think, peer review, but it's hit the, everybody's got a very preliminary look right now at the data that's come out with work that's done by, you can see all the authors here, Steve Horvath et al. Reversing age, dual species measurement of epigenetic age with a single plot. Those of you who read the papers with Horvath and colleagues have been aware now for a number of years of basically epigenetic clocks and what they are. And just to quickly summarize, most of us already know about the epigenome. It's been talked about for many years, but specifically people more recently have noticed, and by people I mean Horvath and colleagues and many others, that the epigenome changes as we age, and it doesn't change quite randomly, but it's stochastic within constraints. So it means that you can predict what kind of changes can occur to the epigenome as a individual ages and as a species ages. And you can basically, use that to determine essentially the biological age of an organism by correlating it with the chronological age, right? So and the epigenetic clock that Horvath is looking at and colleagues is essentially, I think, mostly or primarily methylation based. And they look at many, many thousands of sites. So basically, methyl groups are little chemical groups that gets attached to nucleotides, particularly CPG islands. These are basically repeats of cytosines and guanosines. So CGs, connected with the phosphate, so they're called CPG islands. And those methylation groups, those little CH3 groups, they get attached to the nucleotides. They basically determine whether or not DNA is more compact or less compact. So if you're methylated, you basically can have DNA that has a different compaction state, and that determines whether or not the DNA is transcribed. So, and that changes over time. And normally the methylation status should not change unless, the organism is going through a period of development. And, but throughout adulthood, that status changes, and things can go wrong if the methylation status changes at many locations. And depending on how it changes and what the pattern looks like, you can determine basically the age of an organism. So the more changes there are at particular locations, the older the organism is. Right? So, so that was already determined, and people have been looking at many different species and these methylation clocks. And it's for that reason it's called a clock. So very similar to kind of radiocarbon dating, but here the changes are chemical, right? Rather than nuclear. And they do have physiological repercussions. So it's not just a change in chemical moieties, chemical groups, and nothing happens as a consequence. Things happen that are detrimental because, like I said, these methyl groups affect gene transcription status. So obviously, if transcription changes in the wrong way, aberrantly, then you've got cells undergoing senescence. You have other problems, and so on and so forth. So what did they do in this paper? The reason that people want to, come up with methylation clocks is that they're a biomarker of aging. They're a biochemical biomarker of aging, and they let researchers determine whether or not an organism, is aging, can be aging faster or slower. So if methylation clocks really are telling you what's happening sort of at the fundamental level within a cell, within a eukaryotic cell, how aging is progressing, then you can use that clock to determine whether or not an intervention can have an effect. Right? So if for example, if you give a certain drug to an organism and that organism, its performance is enhanced, does that necessarily mean that aging is slowed down, or does that mean that maybe some physiological parameters are improved, but the fundamental rate of aging is not affected? For example, if you give at the most basic kind of example, if you give a stimulant to an organism, right, it's going to run faster temporarily, but that doesn't mean it's physiologically improved. In all the other parameters, especially fundamentally aging, right? So if you're really going to be reversing the clock of aging, then you want a biomarker that's really going to correlate fundamentally with biological age. And this appears to be the case with methylation clocks aging. So that's been worked out. And what they've done in this paper is taken it a step further. They, as the title suggests, reversing age, dual species measurement of epigenetic age with a single clock. So the single clock is basically looking at methylation status in the genomes of rats and also human cells. And they come up with some equations that basically, correlate the rate of, the methylation status and the rate of aging in rats with the rate of aging in humans, and try to tie those together. And they talk about two parameters that they use. I won't go into that in too much detail, but they talk about absolute age and relative age. Absolute age being basically the chronological and age of an organism that's absolute, meaning if the rat is 1 year old or 0.1 year old, that's the age of the rat, versus absolute, which is a number between, I think, 0 and 1, which would be basically the maximal age of an organism divided by its actual age, right? So if the maximal age is 120 and you're 60, then your relative age is 0.5, right? So they try to essentially correlate rat clocks with human clocks in this paper, using basically a lot of methylation clocks, a lot of statistical analyses. And then they apply it to a treatment, which is basically transfusion of young plasma from young rats to old rats. And I think, not 100%, it's been a while since I read this, if they apply the plasma fractions to human cells. But let's scroll down here. So they look at a lot of tissue samples in rats. So they look at the N is 593 tissue samples, and then they had human tissue samples of 850. And they have 6 clocks. And what that means is that they have a rat pan tissue clock, meaning that they look at methylation status and it's aggregated across many different tissues. And then they have specific clocks for brain, liver, and blood. And then they have 2 epigenetic clocks that basically correlate both, correlate human age methylation changes with rat tissues. I think those are human rat pan tissue. We'll get to that later. Okay. So they established these clocks, which is going to be very useful for many other researchers, but then they apply it to this HPE, which is heterochronic plasma exchange, which is very related to, I'm going to stop sharing here because I'm just talking, which is very related to basically parabiosis studies. So parabiosis, as we've talked about, are you moving the paper? I can only see the top of it with the credits. Yeah, I didn't really move it. We were still on a text, so so I haven't gotten to the data part yet. So there's, I believe, 6 or 7 figures, so we'll scroll to that. So the parabiosis studies are essentially hooking up the circulatory systems of 2 animals, and there's it's not perfect because number one, the animals are under a lot of stress. You basically have them sutured together, and so there's some ethical considerations there. I don't even know. I don't think they can actually be performed in parts of Europe. They are done in the United States. And the other problem with parabiosis studies is that you only get 50% blood, circulation, because you have, and maybe even less because you have a smaller young animal with a larger adult animal that's hooked up. But with heterochronic plasma transfer, they basically can transfer as much plasma from a young animal to an old animal. And earlier studies showed that it's most likely and most probably not because you're not getting a rejuvenation effect. And that's one thing I didn't mention, is that these studies have shown a rejuvenation effect. And this effect in the old animals is not really due to the fact that you have younger organs in the younger animal that are improving the functionality of the older animal, kind of acting as a backup system. But it turns out that there's something in the blood itself, and particularly in the blood plasma. And that's really, really cool, because that can be used as a potential treatment if you're actually targeting the root causes of aging. So in this paper here, they not only established these 6 clocks, laid the groundwork for them to be used by other researchers, but then applied these clocks as a measurement tool to these heterochromic plasma transfers of, from one animal to the other, and then basically looked at tissues and looked at basically not only the methylation status to see if they actually did reverse the aging clock of aging when it comes to the methylation status, but also they looked at physiologic parameters as well, which is really cool. So they did both. So let me go back and share my screen. And one thing I'm going to mention is there will be, I don't know when it's announced yet. Maybe we'll mention this again at the end because I didn't mention in the beginning, but there will be a AMA, Ask Me Anything, I think at some point at the futurology section of Reddit with one of the authors of the paper. So someone there is, Steve, I forgot his name, somebody there is setting that up at some point, correct?
At some point, yeah. Although at the moment, Harold Catcher, who's the PI of this paper, is a little bit reticent to say much in public because we were hoping to have him actually on the Journal Club today, but understandably at the moment he says he doesn't want to risk it because they're still in the process of like dealing with like legalities and IP issues, and he didn't want to come on and say too much. But yeah, the intention is that Reddit will eventually do an AMA, hopefully in unison with us. Maybe we can get Harold to come on when he's a bit more comfortable and talk about it a bit more. That'd be cool, right?
That would be very cool. So, so consider this to be kind of a whetting your appetite. And I'm scrolling through here, and the figures are on the bottom. It's a little because this paper is pretty much almost ready for prime time. So let me scroll. It's all the figures are not your standard position. They're at the end. Hopefully I won't have to bounce around from one section of the paper to the other. I need to look something up. So I'm just going to try to stay to the back with this paper. Okay. So the first figure, I'm not going to really go through this math here, but essentially this is a correlation between the age of the samples. So either, these samples are from humans, so the scale is from, basically it's a regression analysis of DNA methylation age or status on the y-axis with the chronological age on the x-axis. And this is both rat and human. And these are different tissue samples. And this is showing that you have a pretty strong correlation between the methylation points that are being analyzed and the chronological age for most of these samples here. So both, this is rat pan tissue, rat brain, rat blood, rat liver, and then basically correlating rat pan tissue, human and rat as well. And they do this for, so these are all the different tissue types they look at. So there's a key here from adipose, blood, bone marrow, brain. So this is for the pan tissue plots. So they're looking at a lot of different tissues, and then the other ones are basically just looking at specifically one tissue type. So this is basically this cross-validation study of the 6 epigenetic clocks, and 2 of them are actually a correlation of human and rat. So basically, the goal here would be that if you do now experiments on a rat and you correlate it with how do I open this? Whoops. I clicked on chat, but it did not open. Put some problems here. Hold on a second. Messed up. My chat window is not opening up, so I can't see any chats.
Oh yeah, there's lots of chat. Didier is. Hang on.
Oh, here it is. I have to stop sharing. One of the problems is that we know about nothing about the product itself, I think. What is really in the compound? Compound. So by compound, you mean the blood plasma?
Yes, well, if I can say just a few words, then.
Yeah, go ahead.
I'm sorry to say I'm very, very doubtful about this article. Well, I mean, the article, I know that there is a lot of enthusiasm, but there was no test at all for the rats to see if they are living longer. So I don't understand, you know, this is the classical thing, does it work or not? And so this test, the test that was made with 3 times 6 rats is already now, I think, almost 2 years old, or at least it was end 2018. So it was perfectly possible at this time, well, maybe not to sacrifice just 1 or 2 rats, or at least to start again a second test with rats. And so this guy catchers told in a blog with Josh Mitteldorf, who is very enthusiastic about this, that it was not possible because of the lockdown, but this has nothing to do with the lockdown. The lockdown is just a few months from here, and the test was finished more than 1 year ago. So I'm sorry to say that I don't understand why there is so much enthusiasm about this article if the, let's say, elementary thing to test if it is working is not done.
Very valid point. So, so you jumped way ahead into the physiologic parameters, and of course, one major key physiologic parameter that was not measured, obviously, is longevity and lifespan itself. So, they did physiologic parameters for of neurological nature through a maze, for example, and they looked at senescence markers. But sort of the gold standard, which is, well, do these animals live longer, was not done in this paper. It would have been nice, I agree. And then we could have, that's something that could be asked. Oops, I'm going to take the chat, right? I'm going to go back to the chat. And then we'll hold off until we'll do little pauses between figures. Apparently, Steve says it's being done currently. Okay, so that's something that could be asked to the authors themselves at the AMA, because I don't know the details of when they're going to basically be doing the test or so that's something, unfortunately, we cannot answer at the moment here because we don't have one of the authors here. So I can't ask them specifically. So yeah, but that's an important point. But just to kind of backtrack a little bit, these, no doubt, if they don't do it, somebody will do the lifespan assays on these animals with because this is a very promising therapy, and not only is it a promising potential therapy, but people have already been using this on themselves for a long time, self-experimenting, and I'm not talking about the recent trials that have been happening in California in the past few years. I'm talking about experiments going back decades. So so whether or not, and there's many, there's anecdotal evidence that it seems to improve physiologic parameters, but does it improve lifespan itself? That is yet to be seen. Okay, so what did they do with these methylation clocks, and what sort of parameters that they look at here? So they looked at the methylation age versus relative age using these clocks on a number of different treatments. So they did plasma fraction treatment, and then they looked at well, does the relative age, using these methylation clocks change? And then they have various, it's in the materials and methods, the parameters that they used for how often they injected. And I don't have the number right off the top of my head here. Because I don't really want to scroll back and forth. This paper is available to everybody. So they used, again, young plasma and injected into old rats several times, and then they looked later on, I think months later, and we'll see in other figures they actually did many time points and looked at various parameters. But looking at various clocks here, so DNA methylation age, pan tissue, and treatment condition. So this is old control, so nothing, basically probably old plasma into old mice, young control, young mice, and then old treatment, so old mice with young plasma. And you can see that from these bar graphs here, that at least using these clocks, when they looked at treatment conditions for methylation status, they had changes pretty much across the board. The one thing that was a bit aberrant that they've mentioned in the paper is that in the brain, the methylation status didn't seem to change significantly, which is they looked at hypothalamus. And the question is, let's see, is this human, and this is rat, I think. Yeah, human, rat, pan tissue, and brain. So they gave some explanations as to why they think it's not really changing, but it's not really 100% certain why. One reason. Yeah. Okay. So stop sharing. So we have some more comments. Okay, so these comments are still related to the life event. So we'll I'll share the screen back there. So yeah, so right now the focus of this paper again, I'm not going to criticize them for not doing the lifespan because it is what it is at this paper, and no doubt there's going to be a follow-up by them or somebody else, because again, this is very promising data. So somebody's going to really want to see if longevity is impacted. But looking at many tissues, it's pretty clear that using this epigenetic clock analysis, using several different versions of this clock, rat, and also looking at human tissues, you have a dramatic effect when it comes to the changes of the methylation clock. It seems to bring it down or make it equivalent to the young control animal. And now the question is, well, okay, so you have this change happening at the methylation level at many thousands of different locations using these plasma fraction treatments. Does this really correspond to anything meaningful when it comes to aging? So they kind of start to build up on, they do different analyses in these rodents. And they basically, they start off with, organ function, and then they go into more performance-based tests. So they do a lot of different tests on these mice. So this is days post-treatment, so post-injection of plasma. So, and I believe they only injected it in like two different, so I'm going to pause here and ask, like, my question here is how, what I think could potentially be really cool is that this treatment might not need to be, if this is really like an epigenetic kind of a switch, maybe you don't need to do this very frequently. If you do this only once or twice, is that sufficient, to reset the clock in a lot of these tissues? And, the suggestion here is that might be the case. So, you just basically, change your blood plasma at two different time points and then worry about it for a number of years. So they look at different parameters in the blood, so different basically blood metrics that correlate with a young mouse. So we won't go through all of them. So, bilirubin, which is a function of basically liver health, for example, cholesterol levels, total protein levels. So old rats are in red, treated in orange, and young rats are in blue. Some of the blue ones start going up slightly, and that might just be because, well, the young mice might be aging because, it's the rats, so it's 150 days. but you could see basically the orange over a period of time, days post-treatment, starts to drop to levels that in some cases overlap. And, I don't know, there's no error bars here, so I don't know exactly what the overlap here, but you could see a trend happening where these levels are, so high density lipoprotein levels, which are basically, a good carrier of cholesterol, go up and match young mice. Overall cholesterol levels go down, glucose levels go down and match young mice, and so on and so forth. So basically, in pretty much all of these blood parameters that they look at, you over 100 to 150 days post-treatment is when you start seeing this this total reversal. And I don't know why it takes 150 days. So that's another question, right? So if you do this plasma treatment and you get a change that happens rapidly, I don't know why it takes that many several months to do, for these interventions to have these physiological effects. Maybe there has to be some tissue turnover, some homeostasis before that can basically look like a young mouse. Okay. All right, so I'm just kind of pop back out here to kind of look at if there's any chats that are more specific to the figures, and the answer is no. So, but I'm really excited about, at least these physiologic tests that are being done here. Yes, very tippy tip gold standard has not been done, which is longevity, but everything else leading up to it seems to be very promising. Have this figure assessment of plasma fraction treatment of cognitive function. So this is a learning test. So this one is really cool because it's it's an actual performance test. Oops, sorry. Actually, let's figure out this touch screen. Okay. And I don't know the exact protocol of this maze test. So it's a latency. So basically, these mice, there's like a lag before they start to learn how to basically traverse this maze, and the latency in seconds is on the y-axis. And this is days post-treatment. So they basically run this test on a first month, second month, third month, and fourth month. And basically the mice learn, the curve on these plots is how quickly the mice learn, and the latency should drop, right? So in a young mouse in the blue, you could see that the mouse is picking up much faster versus the old mouse, which is learning but much slower. And it gets a bit better as you go from first month to second month, third month, to fourth month, which I believe is kind of long-term memory for a mouse. So if we scroll up, to, so you could see that starts at about 100, and then third month it starts at 80 for the young mouse. But the interesting thing is this orange curve, which is essentially the old treated mouse. So you could see it even in the first month, the mice are improving much faster in their learning. Second month, the improvement is more apparent in basically the beginning of the maze test, and you could see that it's much more pronounced by the third month and fourth month. It doesn't quite get to young levels, but it's significantly improved over the older mouse. So the memory functions and the performance in this this learning and memory test for the rats seems to be quite significant, which is interesting because they didn't seem to find a statistical correlation with the methylation clock, with the hippocampus. So they looked at hippocampus, and what else did they look at? Hypothalamus in the mice. So with treatment of blood plasma. So they speculate that it's not clear why where the improved performance is for memory. It could be that by improving all of the other physiologic functions of the supporting organs, this might be indirectly somehow improving brain function. That's not directly attributable to methylation status in the particular neurons that are involved in memory formation in the brain. But again, further experiments need to be done to confirm that. So I'll take a little break here, stop sharing. So DDA posted a very enthusiastic post of Josh Middledorf. So, okay, so.
A little bit too overly enthusiastic, I've got to say, Oliver. Josh is very sold on the whole programmed rather than damaged theory of aging, and he's very enthusiastic about this.
Yeah.
I've got a few observations about the paper, which I'm going to save to the end. Yeah. But I'm going to say that, yeah, it was a little bit overly excited about it. This is very early stage as far as I'm concerned, and I'd like to see it replicated.
I'll give you, you're in, I think we're both in the same boat. I have my own caveat, which is that, I am in the camp, squarely in the camp with, of non-programmed aging, which Leonard Hayflick also is in that camp. And I do note that in this paper here, it was mentioned that this seems to point to there being a certain, programmed function, because, that the methylation status is that the aging, that the biochemical effects are very similar between species. Therefore, it's programmed. Well, to me, that's still a physics issue, because I mean, if you have two different mechanical devices built out of iron, they're both going to rust in the same way, and that's not really programmed in there. It's just it's a consequence to me of everything is programmed with nucleic acids, and everything uses glucose and everything. So of course things are going to oxidize and be altered in the same way. So that's
for me, it's kind of like saying cellular functions, which are in a way a program, because they do set predictable things. When they receive certain stimuli, that's programmed as well. But that's really sort of stretching things as far as I'm concerned. About the biggest evidence I would suggest that there is such a thing as programmed aging in some species is the example of the salmon. Now that's literally got an inbuilt self-destruct system in it. But in humans, no, I don't think so. I think it's what David Gems is talking about, who is a researcher in the UK. You may know him, Oliver. He talks about the concept of quasi-program. So just because it looks like a program doesn't necessarily mean it is a program.
And I would say, even with the example with salmon, I mean, the salmon are, they basically wreck themselves through releasing a lot of cortisol and a lot of other things to basically spawn. So it is an accelerated wear and tear and aging. And in that respect, it is programmed. However, if that wasn't happening, they would still age, albeit slower. But the reason they do it is so they can successfully spawn. If I can give kind of a very crude analogy, it's kind of like a souped-up race car, where you basically use nitrous oxide to basically over-rev your engine, so you can basically win the race at the consequence of wrecking your engine. If you basically do that too many times, you won the race. Right, but I wouldn't really call that wear and tear programmed. It's that wear and tear is still happening, regardless. But the end point is basically replication and reproduction, right? So that's the species manages to do that. But anyway, that's a chat for maybe we'll have a population genetics chat. I got my own opinions on that. Okay, so they look at oxidative stress as well, and they look at enzymes like superoxide dismutase, catalase that are are there to basically scavenge free radicals, and they also look at things like MDA, which are a consequence of oxidation. So I think oxidized, I want to say fatty acids, but I think it's, what is MDA? It's lipid peroxidation, right? So, so they do see a very, so they look at, I think they look at, this is in the blood, isn't it? Plasma fraction treatment. I think this is looking, I think these are derived. I don't know where they got these tissue samples. Well, they say, oh, okay, right there. Brain, heart, lung, liver. Okay, never mind. So you could see the old levels of, so when they look at old tissues, which is in red, of MDA, which is an example of tissue oxidation that goes up, but it's quite dramatically decreased in young. And other factors that are involved in reversing oxidation or preventing it are also inversely elevated in old treated versus young, as you can see quite clearly in these graphs. Catalyzed function, it's not as significant in some tissues, but it is in others, like liver and lung, for example, and heart, and also superoxide dismutase. So, so it's really cool to see that happening with, because I mean, for a long time, there's been this controversy about correlating oxidative stress with aging, but you see this effect also with oxidative stress, that it tends to go down in these organisms. And then looking at inflammatory markers, so inflammation as something that's really that's a consequence of senescent cells. So interleukin 6, IL-6, TNF alpha, tumor necrosis factor alpha, NRF2. So all of these things that correlate with oxidative stress. If you basically, I mean, just by looking at these graphs, so this is, let's see, what is this? Days after treatment, I think. So blood levels, regular intervals through a 155-day period of the experiment. So this is, I guess, 0 would be at the start of the experiment. So when they started, I guess, just before they injected, or maybe right after they injected, and then days afterwards. So you can see orange, basically 4 days in, it's still pretty high, and then 8 days, it drops pretty low, and it stays low, pretty low, maybe, there's a little bounce up after 90 days, but it stays low all the way to 155 days. So does TNF alpha, and NRF2, which modulates inflammation and oxidative stress, that goes up. So it's an inverse correlation there, except for lung. And I'm not sure why that doesn't happen in lung. But a very significant effect in inflammatory factors, which correlate with senescence. And then looking at senescence a little bit more directly, they look at basically beta-galactosidase staining, which is beta-gal is a it's an enzyme that is apparently expressed in cells that are senescent. So it's used as a marker for a long time. And you basically use a chemical dye to test for its activity. So beta-galactosidase will convert something, a compound, and give basically a blue color. And they don't have this as probably maybe in supplemental. If maybe I'm mistaken, they might have they might have this quantified. But basically, looking at brain tissues, treated old rat, young rat, liver, heart, and lung. I'm not sure if lung is is different, because I don't have the numbers here. It might be. I really don't want to scroll all the way up to take a look, but just by looking at brain and liver, it seems that the numbers are, that you have far less senescent cells. Brain is kind of interesting because, let's see, I don't know what part of the brain they're looking at here. Brain is a big organ, right? There's many different parts. So I'm not 100% sure. And it's interesting that, they showed earlier that it didn't correlate with the methylation status in the brain, yet you had less senescent cells. But that just could be that, I don't know what cell types these are, right? Are these neurons? Are these glial cells? Are these, not really sure. But the senescence markers, if we just look at liver looks pretty clear. The senescence markers are dropped. So a lot of physiological parameters seem to improve. Second infusion at 95 days. Thank you very much. So TNF and I will have dropped. So they do a second infusion 95 days. So okay. So the bottom line is that something really positive is happening with blood plasma transfusions. What factors are responsible is as yet unknown. There's been some controversy in the past with potential isolation of factors that may or may not, be the key factors in having these effects. So it would be really, really interesting to know really what component of blood plasma is doing this, right? Because there's a lot of stuff in blood plasma that that could be playing a role. And it'd be interesting to see, and I don't know how many different factors there exactly are, and if and it'd be interesting to set up some experiments here. Yeah, so TGF beta, Steve says, is a likely suspect. Reduction of which rejuvenates is shown by Convoy. So maybe there's some inhibitors of TGF beta. It'd be interesting to look at different fractions of plasma, if that's how possible that is. The interesting thing is, I'm not a clinician, but they mentioned in the paper that these types of plasma transfusions are actually way easier to do in humans than in rats. So you've got this potential for a treatment here that is actually easier to do in humans because we're bigger, we have more blood, and the transfusion technologies have just been much more well developed for human to human than for rat to rat. So it might be, and the fact that people have already been there's already a protocol in place for lots of blood transfusions taking place. It seems to me that doing these types of experiments in humans, in healthy humans that are, blood typed and matched, the barrier to doing these types of experiments with elderly and young participants should be, I'm not a medical doctor, but should be pretty low, just because, it's very well established, and many millions of people have undergone, transfusions, of various blood products, not just whole blood, but also plasma and platelets for many, many, many decades. So the only thing different here is that you'd be basically age, using different, you'd be using, the variable would be age, essentially. If this really can have such a profound effect, it should be relatively straightforward to set up these clinical trials. So that's my 2 cents on that. And of course, the follow-up would be to do obviously longevity studies on these rats to see if they actually do live longer. But the fact that many physiologic parameters are altering for the better, certainly not for the worst, but significantly for the better, is also extremely promising. So it might be a case here to start clinical trials with that in mind and try to backtrack and find out what are the specific reasons for this working, after the fact. Especially if you can basically set up these types of experiments clinically with a specific disease model, a chronic, a disease state that is directly related to aging, or is it driven by aging? And there's pick, right? There's no dearth of chronic illnesses that are, driven by aging, type 2 diabetes, various, heart conditions, dementias, so on and so forth. So it, one way to kind of get around the anti-aging bias of, at least in this country with the FDA, is to basically pick several different, disease models that can potentially be affected by this, young plasma transfer, and start those clinical trials. And then, with the hope that this is going to, generally impact the root causes of, or at least one of the fundamental drivers of aging in eukaryotic cells in a metazoan, which could be change in epigenetic status. Yeah, correlating with reduction in epigenetic with senescent cell burden for me was the most interesting thing. Steve says, because age reversal is total non-starter with the FDA. And I say FDA because, obviously I'm here in the US, I'm US centric. I don't know how, how many countries are there in the world? 150 something, 200, something like that. As many countries as there are bones in our body. But certainly every country has their different ways of doing things. So maybe an anti-aging version of this could be done in a different country. And in this country, we can focus on chronic disease reversal. But it'd be really cool if that type of trial will take place. And I really don't know what major hurdles would be, because you have a lot of patients undergoing for various conditions, continual blood transfusions all the time, being on dialysis machines. So, there's setups all over the place to do these types of trials. So it's, I think it's just a matter of finding the right people, especially the right clinicians who are wise to the data that's out there right now, and convincing folks to start these trials. So yeah, demonstrable disease modification, as Steve says. So yeah, and that was one of the papers that came out, or a couple papers that came out from Kirkland et al. From the Mayo Clinic, where they basically, not this, but they were looking at flavonoids, and they weren't doing anti-aging, but they were looking at a disease model that's driven by aging. I think they were initially looking at idiopathic pulmonary fibrosis. So, so yeah, Sven says plasma can transfer infectious diseases. Safety aspect, that's true. But obviously, plasma is screened for it, and many, I don't know how many hundreds, probably millions of people do undergo blood transfusions every year. And yeah, that is a risk. Just as an anecdote, a side note, when I said earlier that there was a researcher, people have been researching blood transfers, young blood into old organisms for a long time. There was a famous, I think, researcher that was doing this to themselves in going way back in the early 20th century. I will remember his name later, but I think anecdotally it was working, and then he passed away because the blood was infected with malaria that he used at some point, and that was that. And so yeah, so transferring infectious diseases is obviously a risk, and obviously the optimal solution would be figuring out what factors are involved and having them synthesized, right? So, and then yeah. Exactly. So Steve says, Ronald Kahansky from the NIA said last year that treating aging is a non-starter, but if it's reversed or reversal of age-related disease, then great. So yeah, I think we have a long list of chronic diseases that could be impacted potentially by this treatment, if indeed, as we've been saying, and as the data suggests, that aging is primary driver of many of these diseases. So EDA, yes, you can say a few things since we are in our discussion section right now.
Okay, thank you. So, yeah, sorry to say, but you know this classical sentence when something seems to be too nice to be true, it's too nice to be true. So now a few comments. So we had 18 rats. We had 12 old rats who were following this treatment. Well, 6 were following this treatment during 6 months, and 6 were only to be tested. Okay, and the normal life expectancy of a rat is about 3 years. So almost statistically, you should have 1 or 2 rats dying already. So we don't know anything about the 12 rats. Was a few were a few dying or no? Okay. The second thing is, okay, they took, if I understand correctly, what I see at the end, 17 times they took a sample. So I suppose they took blood, and they had beautiful results all the time. And so at the end, with all these beautiful results, they still decided to sacrifice, to kill all the rats. Well, just to have the epigenetics test, but for the genetics test, you also can just take a sample. So why not to keep a few of these rats alive, just to prove that the rats are living longer? It's really not difficult. And it cost almost nothing, I mean. And then, okay, I know that what they are saying is that they don't have money to test for longevity. Well, at the moment they say it's because of the lockdown. Before it was because they had no money. Now they are saying that they will test soon. Okay, but they say that they don't have the money to test, but they have the money to create a cream that has beautiful results. So if you take a look at the block, you will say that Harold's amazing effect. All his aging spots disappeared in a week, and also hair is growing back on his scalp, and it seems to have effect to the coordination. You know, when you have this beautiful miracle results with a cream. And by the way, I don't understand precisely the link between transfusion and a cream, but I'm sorry, but all alert signals are wet here.
Sorry,
And
are you referring to a cream here? Is there?
yes, yes. When you take I gave 2 links, one.
Okay.
Two articles of the blog, and then at the end of the blog you have reactions of owl catcher and also the person working with him. And this is you can see it. So we have 2 separate products. One is a very powerful anti-aging molecule already tested on Harold with amazing effect. All his aging spots from his arms disappeared in a week. And then Harold wrote, "I'm not here to advertise anything, but it seems to affect my coordination and had hair grow back on my scalp, and I no longer apply it to my skin." So he's not there to advertise, but he has miraculous results. Even hair is going back and so on. Yeah. So sorry, sorry, I don't understand why is there so much enthusiasm. I would love to be wrong, of course, and I would be ready to pay $10,000 tomorrow to test this thing on mice or rats or any other old animal. It's not complicated at all. It could have been done already, like I said, 1 year ago. Well, I mean, 18 months ago. So if it is so good, why don't they are not doing that? That's it.
That's a question, obviously, for them. You know, and when they get the AA up.
I mean, I asked them already. I had already a few email exchanges, and Harold was saying that I didn't understand anything, and he knew it was working, so it was not necessary to make a lifespan test. That was a few months ago, but now he's saying, "Oh no, it's because of the lockdown, and I will do it later." But okay. There is a very small chance I'm totally wrong, and then we will see. But let's push to have this lifespan test.
Okay, well, somebody at some point may do, and most likely a lifespan test, and that's clearly, from what we've seen here, and as Sven's mentioned here, we can't take organ samples of living rats because they're small, and of course that's going to affect them as well. If,
So
yeah.
that's then technical. I don't understand all these results. How do they have these results? For each 4 days, I suppose they take blood. Otherwise, they cannot. Oh, it's so. So this is possible. And I mean, if you take blood, you can also have epigenetic results, I think. But I don't know. I'm not a scientist originally, so maybe I don't understand something.
Well, they're going to have, for the epigenetics, I think they're looking at particular issues, not just blood. So I think they have to kind of sacrifice those animals, unless they're running a colony of rats that were, I think, parallel to this colony, and we're also doing a longevity study that was parallel. But again, I don't. That's a good question. I could ask. When they have that AMA, I'm going to ask them that as well. Maybe we can have a parallel study for longevity with these rats. And there were some inconsistencies, just to point out. For example, with hippocampal neurons and hypothalamic neurons, they didn't see an epigenetic change, but that was so there was a bit of a paradox there. But you seem to have a physiologic response later during the memory test. So how exactly plasma treatment is affecting the brain positively when it doesn't seem to affect the genome, at least in those regions that are known for memory function, it's not clear. So Donovan Walker says, I don't know if this is the case, but I can imagine reporting significantly extended lifespan could make the paper less acceptable, better than a clean paper that creates interest and opportunities for funding. Yeah, that's, again, that's a question for the authors and DDA already, got the answers he got from the authors. But that's something that, we can do as a follow-up. A lot of times, you get the results that you already have. And then, I mean, there's a couple of different philosophies on this. You can have a paper that covers everything and basically really puts the cherry on top of the cake and says, well, we've done everything and also they live longer, and that's fantastic. Or you can have the data that you already have and just publish that and say, well, we'll do follow-up on this later, and that appears to be the case here. But regardless, the data as it's presented here is certainly very promising. So just to kind of reiterate, there's two kind of key takeaway points from this paper. One is that there's a clock that they've been working on, and they've been trying to basically, and it's not 100% certain how effective this, the human to rat correlation is, is how useful that's going to be. But, but that's something that is really promising, because obviously when you do any interventions, if you give a polyphenol to a rat and a rat lives longer, is that going to translate to human, right? I mean, we've already seen this controversy with calorie restriction, right? So shorter lived organisms live by, percentage wise longer if you calorie restrict them versus maybe, already longer lived organisms such as monkeys and apes, humans. So how much of an effect can this translate to health span and lifespan, from a rat model to a human model? So will an epigenome clock that's correlated, between, having a correlation between humans and rats, if you see an improvement in the epigenomic status in using a similar type of intervention in a rat, will this translate the same exact way to human? Obviously that would be perfect if it could, but most likely it's not going to be a perfect correlation. So, but so they established these clocks that researchers can now use. Certainly within rats will be extremely useful. And and then the second part of this paper is basically applying it to an intervention which seems radical, but is clinically kind of a low-hanging fruit already, because we already have established protocols for plasma transfer. So I think those two main points in combination will hopefully drive the clinical studies in this direction a bit faster. So I don't know. That's all I have to say on this paper right now. And again, I know it was mentioned a couple of times in this paper that that the interpretation here is that this gives kind of credence to programmed aging. And Steve and I already mentioned that between ourselves that it doesn't, but we can unpack that further. I don't want to go down that rabbit hole just quite yet. So anyway, let's see. What other questions we had here? For epigenetic aspects, can you do it on a rat alive? Steve, yes, in some tissues like skin, muscle, and blood. Yeah, in blood, definitely. And Sven says not for internal organs. So I haven't worked with rats. Well, I have when I was a technician many years ago, but I sacrificed them, so that's not a good example. Okay. So I'm going to take a breather here and pass the mic to anybody else here who needs to say anything.
I'll take a pitch. As I said in the chat, I think the most interesting thing for me personally here was that the senescent cell burden or presence of senescent cells appears to be reduced when you reset the epigenetic state of tissues. That for me is more interesting than anything else. So I don't know if anybody else felt as excited about that, but there you go. That's exciting. But there's a number of issues with this study, of course, as Didier has mentioned. The size of the cohort, it's only what, 6 rats? Was it 6 rats? 6 or 8?
Yes,
6.
6 times 6 rats. 6 old who were already at the beginning more than 2 years old. 2 times 6 and 6 young rats. And once again, 109 weeks, that's more than 2 years. The experiment was lasting for about 6 months. So normally for the untreated rats, at least if this miracle product is working for the others, 1 or 2 of these rats should die. And certainly when you are making 17 times samples, so it means 17 times touching all these rats. Only the fact that no rat died during the experiment is already very strange, or not mentioned at least.
So yeah, for me, I'd like to see somebody else replicate it, and yeah, they can sign an NDA or whatever, a legal agreement not to disclose, and they wouldn't even necessarily even need to know what's in the compound, just how they've got to administer it. Even better if they didn't know which rat was getting which. So no reason why it couldn't be replicated. So yeah, it's interesting, but yeah. So I think personally, I'm not going to get too excited just yet. Although Victor looks like he's getting really excited. Victor, have you any thoughts on this at all? You usually got something to say about this sort of interesting thing. Well, he's been a bit shy.
Not really a bit. Not right now, actually.
I think the elephant in the room for me, which no one's mentioned yet, is also the dilution hypothesis. This is something that Irina and Michael Conboy have been talking about for a long time. When they did that 2015 paper that showed the reduction of TGF beta rejuvenates, or allows multiple tissues to actually repair better. They also said that they keep saying, I don't know why people keep thinking that it was something in the young blood that was doing it, and not actually the reduction of bad things in the old blood. In other words, it's more about what you take out of age blood than what you put in. And this is the whole dilution hypothesis, that it isn't the actual young blood that you're putting in per se that's important, but the fact that you're diluting the aged blood, and thus pushing down globally the cytokines that are inhibiting stem cell mobility and rejuvenation. So I don't know. Anybody got any thoughts on that? 'Cause
yeah, my thoughts on that is it's if it is just a dilution effect, then you should be able to see that with just saline and not blood plasma. If it could also be not quite a dilution, but it could be an inhibition, right? If there's something in the young blood that basically acts as an inhibitor or binds to factors that are detrimental in older blood, you basically can be the young plasma could be putting in some sort of antagonist, right? So rather than the drop off of an agonist. Right, so those aren't even mutually contradictory. It could be both in the young blood. It could be an antagonist and an agonist that are missing in young blood. But in that case, I wouldn't say that is a dilution per se. It's just the lack of a certain factor that is present in the young blood versus a dilution, which you would think if it was a dilution, then you in theory you should be able to take out that factor and put in some kind of plasma. You should see that effect, I would think, with saline. But then of course you could always counter argue and say that there's some detrimental effect with having just saline. That I don't know. That's my thoughts on this.
So it could be a little of column A and a little bit of column B, folks. So the magic source, there could be still some magic source in young plasma, but equally recalibrating aged blood may also work. Well, we know it works in mice, for example, because as soon as you inhibit TGF beta, for example, using an ALK5 inhibitor, which is what they did in 2015, it reduces the global level of TGF beta, which is a pro-inflammatory cytokine, and we get rejuvenation. So it could be a little bit of column A and a little bit of column B. So the good news is, as Chris Linnell has just pointed out in the chat here, Irina's actually volunteered to work with Harold and see if they can refine the experiment. So the next swing at it maybe tells us a little bit more about that. So that's pretty cool. But
yeah, again, going into a clinician's mindset, I'd love to really brainstorm with a clinician and see if there's ways to do clinical trials, at least in this country, that could be organized in a way, in a medical, I don't know what the jargon is, medical legal way, to basically get the data that we're all interested in, but do it in the easiest way possible. Because, one thing that we all know is that many people undergo blood transfusions for a variety of illnesses and reasons, a lot of times chronically for a long period of time. It'd be interesting if there's a way to maybe set up a trial temporarily with individuals that are already undergoing transfusions and give them blood transfusions that are where the variable is knowing the age of the donor. And then you can test for methylation status basically during the course of their normal treatment. right? Rather than starting a trial like from completely scratch, somehow incorporate a trial where you can basically get, right? Because as far as I know, for medical reasons, it's irrelevant. I could be totally wrong, but it's irrelevant whether you get blood from a donor that's 20 years old or 15 years old, or probably not 15, it's probably age limits, but 20 years old or 30 years old or 50 years old or 60 years old. As long as it's not infected blood and it's healthy, whatever the parameters are used as far as, obviously not infectious agents, but, but if you can age match the transfusions during the course of a therapy and look and see what the influence is. And DDA says, I suppose this was tested already, influence of age. I don't know. I mean, if that was published, certainly not for methylation clocks and status, certainly not physiologic parameters. They probably tested, they might have tested, I'm not sure what sort of tests they've done with age blood. And I don't think there's a limitation to how old you can be to donate blood, right? I mean, they take 70-year-old blood, they take 60-year-old blood, right? So, and there's, if you need type A blood, you get type A blood from a variety of aged donors, right? But if during the course of an intervention, you could, give blood to an individual who's already getting, blood transfusions or blood plasma from individuals. Okay, so DDA just sent me one paper, lack of association between blood donor age and survival of transfused patients. Okay, well, that's an interesting.
Sorry,
yeah.
If I may just, I think also in this article they don't say how much blood or how much plasma was given. And I mean, you take a look, and for some aspects in 8 days, it was like everything was changing. Once again, it's too nice to be true. I really, the more I speak with people, the less I believe it can be something serious. I mean, this is supposed to be one plasma giving really miraculous results. Okay, and they were testing this and they see these beautiful miraculous results, and they don't check later with other old rats, or they don't keep the rats. Really don't.
Yeah, one thing to keep in mind is how much plasma was actually donated, because I think in the rats the whole point was to basically, I think they gave enough plasma to basically.
I think it's not in the article.
Not in the article. Because they do make a point where they say that one problem with parabiosis is that you don't really have enough of a transfer, and I think the point was to transfer transfer all of the plasma or the same volume that was in the rat. And this paper here, which is lack of association between blood donor age and survival of transfused patients, that's Letters to Blood, February 4th, 2016. I have to read that and see, because this is basically just looking at donor blood age and survival of transfused patients. And it's, we're not gonna have time right now to go over that, but that's something worth taking a look at to see, how much, whether or not the metric was a little too crude or not, looking at just overall survival of X thousands of people that are getting some blood transfused from different age groups. But at the very least, that paper does at least suggest that there is no negative effects for at least a small transfusion from people that are older. So these, we could have more of these trials with individuals and see whether or not we have, we can test various physiologic parameters. Well, I have to unfortunately take off in a few minutes because I have to take care of some issues here in the house. I am not in my lab, and unfortunately, I've got stuff to do that is unrelated to science that gotta attend to. But we will. We are planning on having another journal club, and if there's no further questions, I thank everybody for joining me, and we'll take it. I guess we'll take it next month in June. Conference, yes, conference. Oh, I got to mention the Conrad. Totally
Come
forgot.
on, Oliver.
Victor,
Schoolboy
Schoolboy era. Victor, I've been promoting. Yeah, yes, I've been promoting Victor's fundraising here, but we have our own promotions to do, which is we are having our third annual online virtual conference in August. Still August 21st, right?
Yeah, same date. We decided not to move it. We just went virtual.
Excellent. So we're putting together. I think we're putting finishing touches on all of our speakers. We are doing it virtually, so it's our third annual. So a little bummed out about it because we had a venue lined up in Mount Sinai School of Medicine in Manhattan, which is now treating COVID-19 patients and lots of other people. So it's, so who would have thunk, right? A pandemic. So we had all of that lined up, and we had the Stern Auditorium lined up with a capacity of over 600 people, and we will have to take it virtually. But hey, it's going to be just as information packed as always. Unfortunately, we can't meet in the flesh for safety and health reasons. But it will be done virtual. So global it is, just like everything else that's occurring right now. And it will be at the end of August 21st. And we, do check out our website, Life Extension Advocacy Foundation, LEAF, for our upcoming conference, which is going to be really interesting and really cool because I've never done a virtual conference. I've never virtually emceed, and it's
virtual MC, eh?
Virtual MC, yeah. So maybe we can do like a Max Headroom type of like background or something.
And only people of a certain age will understand that reference, but I remember it. But yeah, you can check out details for the conference by visiting lifespan.io/conference. So we've made that super easy. We like that. And despite being disappointed that we couldn't do a third year as a physical conference, because the lineup, the actual venue, as Oliver said, is fantastic at Mount Sinai Hospital. But c'est la vie, that's the way it goes. Yeah. But we are going to make the best of a bad situation. And it's not just going to be like a glorified Zoom call, or if anybody's thinking it's going to be. It's all high tech, all the bells and whistles. It is a really cool virtual environment that we're working with an events company to put on. So I think people are going to be pleasantly surprised by how close we've got it to become almost as good as a physical event. So, check it out.
Is everybody going to be able to pick their own like avatars and skins? Because that would be so cool.
Well, we're going to have a tech demo out in the next few weeks, showcasing some of the things that you can do on it. They're going to be like side rooms and networking opportunities and all sorts. And
I can present as a virtual Klingon. Is that what you're saying?
Oh God, yeah, maybe or as a sentient. Was it a sentient space nebula that you wanted to be once?
Space dolphin.
A space dolphin. Fair enough. He wants to be a space dolphin this
cool.
month.
All right. So you heard it from Steve. If you want to be a space dolphin, you certainly can be, and we will try to accommodate you.
Yeah. Oh, and as an aside as well, you can also take the opportunity right now of getting a free ticket. We are doing a limited access. So what you get is access to the keynote talks on each day, but fantastically, in recognition of these hard economic times, everybody, including the free tickets, also get access to a 3-hour workshop, which you'll recall going to last year, Victor and Oliver, which is the 3-hour biotech workshop with Kelsey Moody, Dr. Kelsey Moody, from iCore Therapeutics. So if you're interested in the workings of how to set up a biotech company from scratch, the trials, the tribulations, the things like employees and structure, there's 3 hours of your time that you really should think about coming along, even if it's just for that. All you've got to do is register on the conference ticket site. It is free, and obviously we'd love to have you along for the rest of it. But if nothing else, come along to that 3-hour workshop. It's well worth your time. So
yeah, and
that's my hot tip.
All kidding aside, it's gonna be really amazing. It's not gonna be a glorified Zoom conference call. Everybody, I'm not the IT expert here. We have folks like Steve and Fatima and everybody else behind the scenes who really know their deal and know the deal and basically corral experts together and put together a really spectacular online event, which is what we're basically planning. So it is going to be, I'm looking forward to it. It's going to be pretty cool. And the fact that I can, MC without wearing any pants that's a huge plus.
Oh boy. Watch this, folks. This will be the bit where he does that and he's totally serious and he forgets. Then he stands up and goes, "Oh, just a minute, dear," and he comes to go and speak to his girlfriend, and then he shows it all. So I hope not.
Yeah,
I hope not. I wouldn't put it past you. I wouldn't risk it, Oliver. My advice: I wouldn't risk it.
Well, I am wearing pants right now. I'm the type of person that is super paranoid about just that thing. I've seen and read about too many horror stories of the worst that can happen probably is my cat is going to jump on my shoulder. That's about it. He's learned recently to perch on my shoulder like a parakeet, and he's taken to doing that, and it's cute and frustrating at the same time, especially when he uses his claws to keep himself anchored.
Exactly. So if you're not wearing pants, cats on your lap, I'm not even gonna think about the possibilities there, just to say that it could be potentially quite painful. So don't try this at home, folks. Always wear your pants. So there you go. And on that enlightening thought, how did we end up here?
I don't know. This reminds me of if you ever watched David Letterman.
Oh, good grief.
The show they always ended the show with "This is brought to you by Worldwide Pants," and I don't know why.
Oh, good heavens! So anyway, on that high note, I'll bid everybody a good evening, day, or night, wherever they are in the world. Do check the conference out, and thank you to heroes, Lifespan Heroes, the monthly supporters, without whom this and many other things that we do would not be possible. So thanks, guys and gals, and we'll see you next time.
Okay. Thank you, everybody.
Thank you.
Bye.
Take care.
I'm going to be